Welcome you all to this cryo-EM course. So, last class I discussed about the about how to prepare the cryo-EM sample. Before that we discuss about how to prepare the negative staining sample. So, first we started with negative staining sample preparation, then we showed how to prepare sample for the protein sample or DNA protein complex or RNA protein complex, membrane protein and so on. And then we moved to cryo-EM sample preparation and how we can do the imaging also. In the meantime, I showed how to do the metal shadowing of the DNA, as well as I discussed a little bit more about the ultramicrotome and different components of the ultramicrotome. Now, in today's class you will see more how we can use this ultramicrotome for sectioning, especially dry sample as well as the frozen sample. Always remember that all these cryo ultramicrotome or today's class or next couple of classes, what I am going to mainly focus how to prepare the sample for sample for tomography or imaging of the any cellular organelles, something like that whole cell or rupture of the cell, and want to see inside the cell what kind of components are there. So, I am going to discuss about all of these things, but the major focus will be that cryo sectioning or the Fib ACM, and then we will use those sample for tomography purposes. So, it will be cryo-electron tomography. So, if you look at my slide, you can see that so most of these techniques again I selected from this particular book. It is called Electron Microscopy Methods and Protocol. So, we will go to the next one, and here you can see couple of chapters I just highlighted here. So, I am not going to give the reference in each and every slides again and again. So, if you just go through that high pressure freezing, current status and the future prospects, or cryofixation by self pressurized rapid freezing, or cryo-electron microscopy of the vitreous section. These three I covered, and again that correlative fluorescence and electron microscope, then Fib ACM for tomography. So, those are the chapters you should focus on that. After you will get my slide or lecture, you just focus on those slides. That will help you to understand what I am talking about, and if you read the book you will get more details and you will get the entire protocol from the from this particular book. This is a very good book about the method and how you can utilize those techniques for your experiment. So, we will go to the next topic, where you can see here there is a two separate techniques I am talking about. One is called the room temperature techniques and the hybrid cryo and room temperature techniques together. So, here you can see that room temperature techniques for the cell, any cell you can consider that is the first you have to do the chemical fixation. For the cryo we are plunging the protein sample or any kind of complex, and then we are getting amorphous ice and protein is embedded there. That is the cryo fixation. Here we are doing the chemical fixation first. So, chemical fixation means, so sample will be dehydrated, then epoxy resin block we have to make it, that is resin embedded, and then we have to perform the sectioning. When we will perform the sectioning, that time we are going to use cryo ultramicrotome or normal room temperature microtome. Then sometime we have to do the heavy metal staining to isolate different organelles and the cell membrane, and then we can perform the room temperature temp. In the other side it is that okay, we can just do the fridge fracturing. So, that fridge fracturing we are going to discuss more. Then you have to drying the sample. When you have the drying dehydrated drying sample, you have to coat with the platinum and the carbon, and then we can use it for room temperature ACM. The other hand, there is a hybrid mode where we can use the cryo as well as the room temperature techniques together. What we can do? We can do a cryofixation, that is your resin block, your tissue, everything will be preserved there, and then you can use the cryoprotection and the freeze in liquid nitrogen, this entire after chemical fixation, and then you can perform the cryosectioning. When you have the cryosectioning, you can use those sample for immunolabeling and figure it out which organelles we are talking about. So, if you just consider that mitochondria has some kind of particular receptor, and then if you have a antibody against that those receptors, and then if you have the gold particle, gold tag, it will go particularly that region, it will interact with this mitochondrial receptor, and then gold particle will be segregated or deposited near the mitochondria. So, it will help you to isolate that what receptor we are targeting or what protein we are targeting inside the cell. Other way is that the cryofixation first. So, you can use the plunge freezing or high pressure freezing. Always remember that for the cell we prefer to use the high pressure freezing, not the normal plunge freezing like single particle cryo-EM. Then we can do the freeze substitution or freeze fracturing. After the freeze fracturing, we have to do the metal coating and carbon coating that we are going to discuss more, or you can just after freeze substitution we can embed this entire item in a resin embedding, and then you do the sectioning, have a heavy metal staining, and then imaging those samples. So, here there is another flow chart, you can see that cryofixation we can use the plunge freezing or high pressure freezing. When we have the sample in high pressure freezing, we can use that again that ultramicrotomy the techniques, that cryo ultramicrotomy, and then we can do the section and we can use it for cryo electron tomography or subtomogram averaging. Other way is that when we have the fixed cell, we can use the Fieb milling and rupture the cell membrane and go inside the cell and perform the tomography. And normal when you have the purified protein sample or DNA protein sample, RNA protein complex or membrane protein, we can use the plunge freezing and we can use the single particle analysis that we already discussed. So, we can perform the micro ED also, that we did not discuss in this class. In the later maybe we can talk about this micro ED in the next course. Again, after high pressure freezing, Fieb milling is cryo Fieb milling is one of the possibility, and do the cryo imaging. Otherwise, you can do the freeze fracture, then etching and then coating with the metal and carbon and perform the cryo ACM. Okay. So, now first come to the high pressure freezing current status and the future prospects. So, if you come to this one, what it is telling that any sample like optical microscope, when you are using, somehow we have to fix the cell or the specimen on the glass slide, and then we are using that glass slide for the imaging. Here also cryo immobilization is an important technique. So, cryo immobilization is the optimum techniques to preserve the ultrastructure of the biological specimen, or more generally soft condensed matter. However, the freezing of the sample thicker than a few micron, the most important term is this few micron, without using the anti-freezing agent and a ambient pressure leads to considerable ice crystal formation and distortion of the biological material. What does it mean? So, if you just consider there is a cell, and inside cell there is a different kind of organelles are there. Now, if you do not freeze the sample properly, that means if you just use the normal cryo freezing, that you have a grid and you are adding the cell and then quickly plunged into the liquid ethane, what will happen? We are expecting that liquid ethane will absorb temperature very fast, but it will absorb temperature very fast from the outer part of the cell. So, that will be outer membrane or the outer plasma membrane, but what about inside the cell? There are so many other liquid and the dense objects are there. So, it cannot absorb the heat very fast inside the cell, because cell is coated with a lipid membrane. So, then the entire process will be slow. When the entire process will be slow, inside this will form a different kind of crystalline ice, that may be hexagonal crystal or cubic crystal, and that will going to, if there is all over a crystalline ice, that is going to hit the cell organelles, it may be the mitochondria, it may be the mitochondrial membrane, or it may be the Golgi or endoplasmic reticulum, and it will damage those organelles. So, if we try to image that sample, we cannot get the proper image because of the ice crystal formation inside the cell and because of the very slow freezing that ice crystal is forming. Okay. So, that is here it is mentioning that we are not using any anti-freezing agent. Within cell, anti-freezing agent insertion is very difficult, and the ambient normal pressure when we are trying, it will form the crystal and distortion of the biological material. Such sample can only be frozen adequately if intercellular anti-freezing agent are used or under high pressure. So, intercellular anti-freezing agents are normally not incorporated by the living cell or the organelles under a physiological condition or a problematic concern normal physiology of the cell. So, we cannot use the anti-freezing agents normally when we are trying to freeze. So, then best way it is that high pressure freezing was developed to overcome this drawback and allow adequate freezing of native biological sample of up to 200 micrometer in thickness. So, generally we are talking about a very thin sample in 100 to 200 nanometer. Here we are talking about 200 micrometer thickness of the cell or tissue that we want to freeze, and we do not want to damage the cell by because of this crystal formation. In that cases we can use the high pressure freezing. How it is working? So, generally we are using a very high pressure, that is 2100 bar need to be applied to the sample within a very few milliseconds, followed by immediate rapid cooling to prevent the introduction of artifacts caused by the high pressure. So, we are going to create a very high pressure, that is 2100 bar for a very small amount time, a very few milliseconds, and try to freeze it immediately. So, that high pressure freezing becomes a state of the art technique, contribute to the elucidation of numerous scientific questions which cannot be answered using the room temperature preparation techniques like chemical fixation. So, there is no dehydration, sample is in hydrated condition, and that basically helping us to get that high resolution details of inside the cell. Now, what kind of instrument we can use it? So, there are couple of names of the instruments are there, that is EM HPM 100 or EM PAC2 which is coming from the Leica microsystem, or HPM 010 or HPF compact 02. So, what is the take home message? Rapid increase of the pressure near the sample area, typically up to 2100 bar or 2000 atmospheric pressure. This pressure will applied within a few millisecond. The point is that this is the important part, that few millisecond to ensure that water in the sample does not have time to crystallize. So, it should not form crystalline ice or it should not crystallize. Now, there should be a continuous cooling. So, liquid nitrogen flow will be there for the rapid cooling of the samples simultaneously with the high pressure. Pressure plateau and release. After reaching a maximum pressure, the pressure is held constant while cooling continue, and once the vitrification is completed within a few milliseconds, the pressure is gradually or abruptly released. Okay, how it is working? So, now this is the instrument where we are keeping, this is the major instrument where you have a binocular, you have a touch screen where you can control many parameters, you can insert the liquid nitrogen dewar through this one. I am going to show you little bit more about that when I will go to that particular part, that I have a small video from the Leica where you can see how we are inserting the sample. Now, the most important part is this particular part. This is the lower half of the cylinder, total length is only 2 centimeter, where sample can stay here. So, this is the middle plate, this middle plate will come and fix somewhere here. When this middle plate fix here, in the next part, this is the area where we are going to add the sample. The sample will be there, a sample container it will be there, where the sample will be there, and then that particular part will be locked, and then that will place somewhere here, and then this is the upper half the cylinder that it will come from the machine and it will lock particular here. So, first the lower half the cylinder, then this middle plate, and then there will be a upper cylinder. So, it will be something like this, it will go. So, middle plate, lower plate, middle plate and upper plate, this entire thing it will go inside the instrument. So, in the next step here you can see again, this is the half cylinder here where it is located, this is the upper plate it will place, where this is the place where we are going to add the sample. So, this will place here. Now, this is the sample carrier, what I already showed you last slide, that okay, this is the first carrier here, we are adding the sample here, and then we are blocked that entire part. When that is blocked, this will placed somewhere here, and then upper part it will, upper cylinder it will come and fix this entire thing here, and it will go inside the instrument. So, we will just play a small video that will help you to understand how this entire thing is working. Only that sample loading and sample insertion we are going to talk about. So, this video is taken from the Leica website. This is the Leica instrument, and and that is the most prominent instrument nowadays. People are using this particular instrument. So, here you can see this is the instrument here, what we can use it for imaging. So, here you can see liquid nitrogen we are inserting here. So, if you just consider that, so we are inserting the liquid nitrogen. In the next step, we will just go and this liquid nitrogen will go inside, and then we will focus entire, we will insert that liquid nitrogen, and then using the binocular we will just focus the entire area properly. This is the screen display, whatever parameters is required we can fix those parameters properly. Then using the binocular we will adjust an alignment rest of the part, and then we will go for sample sample loading stage. So, now you can see there is a area where we are adding the sample, then there will be a cover, on top of that we will put the cover, excess sample will be blotted using a filter paper, and then there will be a setup, it will come pick up complete cylinder will go inside the chamber. Now, again there is a sample holder, we will put that sample will added here, it will covered with the covered slip, or the sample will be covered properly. Then instrument it will half cylinder it will come, pick the entire thing and go inside the high pressure freezer instrument, and this will so there are different types of sample holder is there that you can place it one by one and you can image those things in a different way. So, we are not spending more time here, we will go to the diagram it will help us to explain. So, whatever I was discussing here, you can see this is the cylinder plate. Here there is a spacer which or tape or other spacer where the middle plate, this is the middle plate, and this is our sample holder that we are going to keep it here. On this sample holder we are adding a small amount of sample. If you look at the same particular diagram through here, you can see this is the particular plate of this one, that cylinder plate that we are talking about, the half cylinder, and this is our central plate that or middle plate with a hole where we are adding the sample. Where we are adding the sample, here is the we are adding the sample. If we look at this, how this sample is staying here, there is a platform, and top of that we are loading the sample. After loading the sample, what we did, we covered the entire sample with with another instrument that is holding the sample properly. So, and it is making some kind of cartridge kind of thing. So, here you can see there is a 200 micrometer pressure ring and 6 millimeter aluminum specimen carrier type 1, that it is loading here, your sample is staying somewhere in the central part of this, and this entire thing is going inside the high pressure freezer. Now, when we did that high pressure freezing and do the cell sectioning and then we are imaging, we can see different types of cell organelles inside the cell, and that will be very much clear, and where you can see different types of vesicles also inside the cell, and that is very prominent from this diagram where you can see a large vesicle is spreaded all over. So, you can using this high pressure freezing instrument, you are not damaging, even you can see lots of other organelles outside this cell area, that you can also clearly visualize using this instrument. Now, next, this is again it is showing how we are packing the sample for the high pressure freezer, and this is the cell area where you can target one particular cell, that okay, you are targeting either this particular cell or this particular cell, where you are focusing on all these details and then you are trying to image those samples. This is only possible if you if your cell is not damaged by the high crystalline ice, or the inside if it is not from the crystal ice. So, this is the membrane carrier for this fridge fracture. Now, we are going to talk about the fridge fracture, that is we are going to discuss more in details how we can do the fridge fracture. Fridge fracture is another important techniques, but before going to discuss about the fridge fracture, let us see the self pressurized rapid freezing technique. So, this is one of the another important technique. In this particular technique, we do not need that very high pressure freezing instrument, and that is bit expensive also. In that is the case, we can use this high self pressurized rapid freezing technique, where the self pressurized rapid freezing technique is a cryofixation technique used in cryoloctron microscope to preserve biological samples in their native hydrated state. In this method, sample is sealed inside a metal or a plastic capillary tube and rapidly plunged into cryogen, typically liquid ethane or propane. So, in this particular case, we are putting sample in a very thin capillary tube and then sealed it and then quickly plunged it. When you are doing that, what will happen? When ice will freeze, so water cools and begins to freeze, it expand its size. So, when it will expand, it cannot get enough space because that capillary tube is already sealed. Okay. So, but the sealed environment prevent volume expansion, causing pressure to build up inside the tube. So, we are using this method to create a high pressure during the freezing. So, this self generated pressure, typically several hundred megapascals, helps suppress ice crystal formation, allowing the sample to vitrify more effectively even in slightly thicker specimen than would otherwise be possible with conventional plunge freezing. So, how we can use these techniques? So, here we are showing that self pressurized rapid freezing technique. What is that self pressurized rapid freezing technique? Here you have a pipette where we have the capillary tube. We are using this capillary tube, and you can see using this metal capillary tube, using the pipette tip, we are taking the cell suspension inside that tube. When the cell suspension is inside the tube, one side there is a metal tube which is attached with the pipette, and the other side will just use the pillars and then fix that end completely. So, it should not remove any sample, and the other side will seal that other side also. Now, we will take that metal tube and then quickly plunged into liquid ethane at minus 180 degree temperature. When this entire thing is frozen at the liquid nitrogen temperature, again we will use the cryo ultra microtome and perform the sectioning. When sectioning is done, we can use those thin section sample for the imaging purposes. So, if you go now after frozen, here you can see that intestine region of a nematode, the cryo fixed by SPRF, that is the previous technique we are discussing, that self pressurized rapid freezing technique, and then it was subjected to freeze the entire sample. Then we are performing the sectioning and trying to see inside that cell. If this technique does not work, you can see lot of ice crystal and it is damaging the sample completely, but in this particular case you can see the cells are very clear, there is no damage due to the ice crystal. Even the lipid bilayers is clearly visible from those images. That is the most important part, where you can see the lipid bilayer difference and lots of other components also you can see it from those images. So, if you just look at those cell, again those are the yeast cell, where you can see that Saccharomyces cerevisiae is prepared by this SPRF methods and freeze substitution, and the outer region of the yeast cell, you can see it here, you can see the cell wall, and then if you go inside that, you can see the endoplasmic reticulum. Here it is written that endoplasmic reticulum, this is your cell wall, you can see clearly. Not only that, you can see the cytoplasm, you can see the nuclear envelope, you can see the nucleus, and so you can see this is the nuclear envelope. You can see this bilayer clearly is visible, you can see the nucleus inside, you can see the nuclear pore, here you can see the nuclear pore also in those images. So, if you do a proper sectioning, you can see lots of details inside our cell that will really help without having a very costly high pressure freezing equipment also. There is another example of this self pressurized rapid freezing technique, as well as that cryo-electron microscopy of vitreous section. So, here you can see this is one of these after high pressure freezing, that we applied that vitreous sectioning to image all this different cell. Mostly here you can see that is called that yeast cell. In this yeast cell, if you just after sectioning, you can see this is the cell wall again, this is the endoplasmic reticulum, this is nuclear pore, this is the nucleus, boundary of the nucleus, this is the nuclear pore, you can see it. Not only that, you can see the fine details of yeast mitochondria here. So, this is a mitochondria of the yeast, and you can see all these fine details inside that cell. So, this is a very important technique how we can use it, that self pressurized rapid freezing technique. But then I also used one more term, that is called cryo-electron microscopy of vitreous section. What is that cryo-electron microscopy of the vitreous section? That we are going to discuss little bit more, because because that is how we can use that cryo ultra microtome, because we already discussed about the microtome and microtomy techniques, but how to use that cryo ultra microtome and how to do this sectioning, that is called that cryo-electron microscopy of vitreous section. So, that we are going to discuss in the next class, and including that cryo sectioning, we are going to discuss more about that fridge fracture, and we will discuss more about the fridge fracture, and as well as we will discuss about the Fib ACM in the next class. So, thank you all for your attention.